GHB, or gamma hydroxybutyric acid, is a drug that acts as a nervous system depressant. This drug is also known as liquid ecstasy. It slows down cell signaling/messaging between the body and the brain. GHB is used to produce high levels of euphoria, relaxation, and sociability. Side effects include drowsiness, vomiting, dependence, as well as serious unconsciousness and respiratory collapse. This can lead to a coma or even death. While it can be used for good under certain conditions, it is commonly used as a date rape drug. GHB is a competitive inhibitor to GABA in the pathway it affects. It uses paracrine signaling, which is when cells communicate in close proximity to each other. The normal pathway uses GABA as the ligand. GABA is an inhibitory neurotransmitter. It stops signals in the brain, which results in a calming feeling. GABA binds to the GABA B or beta receptor. GABA B is a G protein coupled receptor. It is part of the GABA complex, which is located in the central nervous system. The GABA complex is made up of GABA A or alpha, GABA B, and GABA gamma. When GABA binds to this, it activates a cascade. In the GABA B receptor, after the binding of GABA, GERK channels or potassium ion channels are activated and calcium ion channels are inhibited. This means that potassium is flowing out while calcium is kept in. The release of potassium causes a lack of communication between cells in the brain. Adenylyl cyclase is inhibited during this process, which lowers the production of CAMP. Adenylyl cyclase is a membrane-bound enzyme that converts ATP into CAMP. CAMP acts as the second messenger in the pathway. When CAMP isn't able to be a second messenger, pKa, or protein kinase A, isn't activated. pKa helps with something called synaptic plasticity. Synaptic plasticity is the ability of neurons to strengthen or weaken connections. Since pKa isn't activated, the synaptic plasticity is weakened. The response of the normal pathway calms you down. This is because there is less brain activity happening. When GHB interferes and takes the place of the normal ligand, the response it creates is a heightened version of the normal response.